Hello. First, I want to thank the organizers for inviting me to present the progress of our SQANTI3 project. SQANTI was published in 2019 as a tool for the quality control of long-read transcriptomics data and has evolved since into an ecosystem that includes different functionalities. Usually, the input is long-read transcript models, although you can also provide just long reads of any transcript model. If there is a reference genome annotation, the quality control tool will map the transcript models into the annotation, classify them into SQANTI categories, extract over 50 quality control features, create diagnostic plots, and also now annotate the models with functional labels. In SQANTI3, we incorporate now orthogonal data such as Illumina reads and CAGE to support the models. We have added now the SQANTI proteins that adapt the SQANTI classification to better describe the variability at the protein level and support proteomics experiments. If you don't have a reference, the new SQANTI no reference script will be able to provide a classification scheme too. Once transcripts are characterized, this information is used by the machine learning tool to differentiate true transcripts from artifacts that are frequent in this data and curate your transcriptome, which then can be used to map back the long or short reads and obtain quantification values. Due to time constraints, I will only talk today about new features, machine learning, and SQANTI without a reference. As you may know, SQANTI classifies long-read transcripts for their similarity to a reference transcript at splice junctions. Hence, full splice matches have all their junctions similar to a reference transcript. Incomplete splice matches misses some of them at the beginning of ends, but the rest are okay. Novel in catalog is when you have a new junction, for example here, that is a combination of existing donor and acceptor sites, while the novel not in catalog has at least one novel or acceptor sites, like here in this example. In the past, our filter looked at the junctions mainly, and we were able to call most of the novel not in catalog as a transcript artifact, while the others, like the incomplete splice matches and the novel in catalog, were maintained. However, we know that incomplete splice matches might be degradation products of true transcripts, and we also observed a great diversity at 3' ends and 5' ends, with sometimes many full splice matching to the same reference transcripts. And we are now willing to make some contributions to take care of this redundancy and remove artifacts from the data. So we are adding CAGE peaks and, if available, also Illumina reads and polysite motifs to the set of SQANTI QC features. CAGE data are not always available, and in case Illumina data are, SQANTI calculates the ratio of short reads around the transcription start site of the transcript model. If the TSS is correct, we expect a high value of this ratio, and if the transcript model is an artifact, we expect the value to be around 1. When we look at some example data, we find that CAGE support is good for full splice matches, it's true. However, for incomplete splice matches, it's mostly non-covered by CAGE peaks, although there is a significant fraction of this transcript that have CAGE support and could be bona fide transcription start sites transcripts. Also, for the ratio TSS, you can see that while full splice matches have a bimodal distribution with most of the TSS with high value, most incomplete splice matches have values around 1 as expected for degradation products, and only a fraction have values similar to full splice matches. When we use this information in our machine learning filter, we observe now a considerably filtering out of incomplete splice matches with an intensity that is dependent of the amount of complementary information used. indicating that we can couple this orthogonal data to SQANTI to better curate the long-read transcriptomes. We now talk about SQANTI without a reference genome, which is another feature. As you may know, long reads are frequently used by researchers to study transcriptomes without having a genome annotation, and we want to create a classification scheme in this scenario. The problem here is that the sequencing provides transcript sequences, and it is easy to identify the exons and alternative splice patterns when the reference is available. But however, when you don't have a reference, this is more complicated, and all the clustering algorithm could identify multiple transcripts of the same gene. Without the reference, it's hard to indicate which are the exons and the alternative splice patterns. In this case, we have envisioned a classification scheme that first identifies the transcripts, a transcript that is the baseline transcript, the BLT, and then describes the rest for the differences to this BLT, like intron retention, exonic fragment exclusion, inclusion, etc. So the way we do this is using Cogent, that is a program generated by ListSem to cluster transcripts into the gene of origin and create a reconstructed express locus that integrates all transcript sequences into one contiguous path. Then in SQANTI, we map back the transcript to these reconstructed express loci to identify fragments. These fragments are first extracted and characterized in terms of the 3 and 5 prime ends, the lengths, GC contact, etc. But also they are processed with SUPA to extract events and obtain the coordinates of these splicing events. This info is then combined to finally annotate the fragments as alternative 3 prime, exonic fragment, alternative 5 prime, intron retention, etc. This fragment annotation is mapped back to the reconstructed express locus, and then is quite straightforward from this point to annotate the transcripts as the BLT of the intron retention of all the other categories. We have evaluated this approach in different data sets, and here we present results for Arabidopsis, where we can see that although some misclassification is present, in general, the misclassification rate is below 0.5. And with this, I would like to conclude my presentation today with acknowledgements to my students and the funding agency, and actually to collaborators that are active developers of this set of tools. Thank you.